So you can already start admitting people. I guess we could start now.
Okay, so JP, if we could share our slides. Yeah, so good morning to everyone. We are live on Zoom and on YouTube. Welcome to the Filipino Science Hub. This morning we have a webinar entitled Cancer Genome Analysis delivered by Professor Chad Creighton from Baylor College of Medicine. So um, next slide, please. So before we move forward, we just have a few house rules for this morning. We kindly request for everyone to mute their audio and turn their video unless there's a question or comment. You can type it on the Zoom chat box as well as on YouTube. Also, may we kindly request for you to not record our session. The full video of the event will be posted on our YouTube channel. For the certificate of participation, we will post a Google form link to issue a certificate of participation for this session. So we will post that on Zoom chat box, YouTube live comments after the webinar. So it will be, the video description in YouTube will be after the session, but make sure to subscribe to PhilSci Hub's official YouTube channel and also like us or follow us on Facebook. So again, welcome to the Filipino Science Hub to talk more about our community. May I have Dr. Jeff Punkin. Dr. Jeff?
All right. Thank you so much, Professor Dindy. Good morning, everyone. I'm Jeffrey Punkin, and I'm the founder of Filipino Science Hub. So I'm actually a practicing Filipino scientist based here in Houston, Texas. So muli, maraming salamat po sa pagsama ninyo sa amin sa isa na namang Sabado ng pwede tayo matuto ng sa isang bago na namang larangan sa sciences. So, but before we actually proceed with the talk, I would like to introduce the Filipino Science Hub to our new audience, to those who are not yet familiar with our organization. So, the Filipino Science Hub is is a volunteer organization that I actually founded back in 2012, and its main intention is to actually promote the cultures of STEM education and research among Filipino students and teachers. And at the moment, this organization is being led by 6 Filipino scientists trained in different STEM areas based in different parts of the world. So we do have Professor Dindy, who's our host for today. She's actually based in Kentucky, Texas. We have Marty Mateo, who is a researcher at the Institute of Plant Breeding in UP Los Baños. We have JP Onya, who is an analytical chemistry and electrocatalysis expert who's doing his PhD at in Finland. And then we have Professor Medjoan Aguila, a professor of chemistry at UP Los Baños. And last but not the least, like Janice Aurelia, who is a researcher, a food science expert at the University of Kansas Medical Center. So, okay, so what do we do at PhilSci Hub? So what our ultimate goal is to actually launch again, a new generation of uh, Filipino STEM enthusiasts. So we would like to be able to encourage the youth to venture into a career in the sciences. And how do we actually plan to do this? There are two components of the strategy. So the first, which is to okay. So the first of which is to empower our educators and help improve their mastery of STEM subject matter areas. So this is actually very important because our teachers are at the forefront of STEM education. So the more empowered our teachers are, the higher the quality of educations that our students will be able to avail of. And then secondly, we also have launched programs that provide research training for students and teachers at the high school and college level. So what the mind knows, the hands should be able to actualize. So and we actually do this not only by training students and teachers in research, but by also bringing in experts from different areas to introduce their respective research areas and also give you guys an idea of what it's like to actually work say like in different STEM fields. And so actually today we're quite fortunate enough to have our very first international speaker. And this is actually one thing that we promise we'll do at PhilSci Hub, that we would like to give you we would like to make you experience a sense of international STEM community. So it's very important, for us to be able to be part of a global STEM ecosystem. And so for us to be really successful at research, we have to be able to collaborate and, and stay connected with experts from different countries. And we are very privileged today to have our speaker, who's actually a researcher at actually one of the top universities when it comes to medical sciences. So watch out for his talk. Okay. And so we've actually been conducting web events over the past 11 to 12 months now. And so, I think like so far we've already had about 26 or 27 web events. And these are actually comprised of teaching webinars. So we train teachers, career guidance talks for students, web lectures and tutorials, like to help enable to help our teachers like master different STEM courses, research webinars, the training courses that we've been offering at PhilSci Research University, and also STEM special topics. And this is actually one of them. So we've already rendered about like 63 hours of expert experience to about 14,000 live participants. So and that translates to about 35,000 training hours already. So we're starting to make an impact this way. But the good thing about this is that all of these web events that we typically conduct, they're recorded, they're uploaded on our YouTube channel. So if there are some contents that you missed, just actually go to our YouTube channel and watch them. They're for free. You don't have to pay for anything. So it's freely accessible to everyone. All right. So for today, I will actually talk about our program on education. So we have recently launched Filipino Science Hub Education Program. And the main purpose of this is to actually train our educators. So at the moment, imagine this scenario. We have about 30,000 teachers who are following our platform. If we get to empower these teachers, and if each of these 30,000 teachers would teach for an average 30 years in their lifetime, and for each year, if they get to teach and influence about like 30 students, you're looking at about 27 million Filipino students impacted over a span of 30 years. And this is actually going to be very significant. If only 1%, say if 1% of that population ends up pursuing a career in the sciences, we will be fairly competitive with our neighboring Southeast Asian countries. So, where we're lagging behind when it comes to the number of scientists like in the country right now. And this is our way to actually address that, to encourage our students to become scientists in the future. And how do we actually do this? So we've been providing, as I mentioned earlier, we've been providing like free teaching webinars to our teachers. So we have recorded webinar, we have recorded training materials that our teachers can avail of. So, you know, watch out. And then we have a couple more scheduled soon. So watch out for further announcements. So like all these are on our website and on our YouTube channel. We also recently launched a modules program. So to actually provide supplemental materials that our teachers and students can actually make use of, you know, uh, uh, as they move on with the new normal mode of teaching. So like, you know, we have actually been rolling out free teaching modules in chemistry, biology, and physics. So, you know, like these modules contain teaching guides or learning guides, prescribed class activities that can be performed at home, as well as problem sets that can actually help in the assessment of the learning. So we roll out modules on a daily basis. So just like visit our Facebook page and our website, you know, for you to be able to access all these like freely accessible materials. And then something new, and this is something that we're partnering up with a couple of huge organizations in the Philippines who are promoting uh, who are actually advocating for the upgrading of the quality of teaching in the country. So we're about to launch our PhilSci Hub STEM Teaching Fellowship. So in this particular program, we will actually be inviting some early career teachers to undergo this training program, you know, where you will be mentored by experts in education, experts in pedagogy, and also you'll also get firsthand training from scientists. So we will train you on how to teach STEM effectively on a practical level. So watch out for this. So we will be announcing the details of this on May 8th of essentially next month, so a couple of weeks from now. So watch out for this. All right, JP, take it away.
All right. Thank you, Doc Jeff. Good morning, everyone. My name is JP, and I am the head of research at PhilSci Hub. And I'm going to introduce today our program, PhilSci Hub Research University, which is a webinar series focusing on scientific research and all its different aspects. And today, we have a special topic for Research University, and it's cancer genome analysis with Professor Chad Creighton. So while PhilSci Research University is our online campaign where we share some of the best practices in scientific research and share our own research experiences when it comes to carrying out a research project. Next slide, please. So PhilSci Research University is composed of 8 mandatory courses, starting from research ideation, literature review, and right now we have finished 5 courses for this, and the next one is coming up next month, and that would be on writing a scientific report or scientific communication. So next slide, please. So the full details of this campaign can be watched from our YouTube launch last January, and all of the details are there on how to join the Google Classroom, because we've set up a Google Classroom for this campaign, and people can go there and watch all the previous courses and obtain a certificate of participation in those courses post facto. And if you completed all the mandatory courses for Research University, you're going to get a completion certificate, and that's also described there in the Google Classroom on how we could obtain the completion certificate. So please kindly visit that, and I will also send the link to the Google Classroom when I send out the certificates of participation for this webinar. Okay. So yeah. So PhilSci Hub is ever present in all of our platforms, mainly our website at www.philscihub.com, where it contains all of our webinars, tutorials, modules, virtual labs, and some special features. And we're also ever present on Facebook, where we have, let's look at it, 30.5K followers. And on YouTube, we have 9.6, and we're even present on TikTok. So please kindly visit our website for all of our contents and web offerings. And yeah, we'll catch you. Yep. So I can hand over the screen now to Professor Dindy.
All right, thank you, Mr. JP. So just a kind reminder, Paul, to keep our audios off during the webinars to avoid distracting our speaker for tonight. So we, just like what Dr. just said, we are so fortunate to have our very first international speaker, Professor Chad Creighton from Baylor College of Medicine. Next slide, please. So just a little bit about our speaker for this morning. He is a professor of Baylor College of Medicine, co-director of Cancer Bioinformatics, Dan Duncan Comprehensive Cancer Center, affiliated faculty member of Human Genome Sequencing Center. So he got his PhD in bioinformatics from University of Michigan, and he got his bachelor's degree in physics, summa cum laude, from University of Idaho. So Dr. Chad is an accomplished scientist, accomplished professor. He has over 300 publications so far. Am I correct, Professor Chad? So we are so glad to have him speak for us about his expertise. So without further ado, may I give the floor to Professor Chad. Chad, take it away.
All right. Okay, everyone can see the slide?
Yep.
Well, thank you very much for that introduction. I'm very excited to be here and to share some of my work and to talk a little bit about my journey and how I ended up doing the work that I'm currently doing. And also to talk about this cancer genome analysis, which is a very exciting field, one that's gone through a lot of changes over the last 20 years, just with this explosion of data and all the opportunities it gives us to really understand what's going on in the cancer cell and to understand cancer at a depth that wasn't possible before. So in my slides, I'll give a lot of history and kind of like a lot of the earlier studies that kind of came before, and then I'll end with some of my own work that's carried out in collaboration with others. So just I was asked to talk a little bit about me and how I ended up in my current role. For me, bioinformatics was not a straight shot. It wasn't an easy, clear, simple path, I guess, from where I started to where I ended up. I did not originally intend to go into bioinformatics. Bioinformatics didn't really exist or wasn't talked about when I was in high school or college. It wasn't anything that was presented as a career option. So it was a new field that sort of came out maybe 20 years ago, and so it's just something that I got drawn into. But my bachelor's degree was in physics. I studied at the University of Idaho from 1992 to 1996, and originally I thought I was going to go into graduate school in physics and be a physicist. And then I changed my mind. My senior year, I decided I did not want to continue in physics. It's a long story how that happened, but I actually took 5 years off between college and graduate school. I worked a couple different jobs, and my first job after college was working in a microbiology genetics lab. I took a biochemistry class from a professor my senior year, and he found out I was going to have take a gap year or more off to take a break from school. he invited me to work in his research lab, and I ended up doing that for 2 years. And this was what's called wet lab work, where it's a lot of a hands-on experimental work, working with PCR and cloning and working with DNA and in bacteria. So I had no knowledge, not much background going into the job, but I really enjoyed it for a couple of years. Something very different. It also got me introduced to molecular biology and a lot of the tools involved, as well as research. But I thought it was, to me, it was just maybe an interesting detour. It's just something it wasn't going to be a permanent thing, that I would just try this for a couple of years and then go into something else. And what I had planned to do was to learn computer programming and become a software developer, because there were a lot of jobs in the late 90s. A lot of computer startup companies were being formed, and they needed computer programmers, and it was an easy way to get a job. And that's something that I took some classes and got a job at an engineering startup company as a software developer in 1998. And I did that for a few years. And so that was working in the computer side of things, very different from the research biology side of things. And I thought that would maybe be my career would be in software development. But I guess I felt like it wasn't really where I was supposed to be. It's kind of funny, but in the year 2000, the human genome was sequenced. It was a draft sequence, but it was sort of the first fairly complete version of the human genome. So before, they just had pieces of the genome that were sequenced, or specific genes, but then the United States government carried out a project to systematically sequence all the genome. And so that draft sequence was announced in 2000. It was on the news, so I listened to it on the radio, on the television, and it's something that just somehow it just resonated with me. It was somewhat exciting. It was like when we went to the moon in the 60s. It was sort of like a new era, and maybe opportunities to learn new things. And I wanted to do something really interesting with my life, so I thought I would go to graduate school. But this time I had an idea of what I wanted to study, or before I didn't have much of a direction to what to study in graduate school. But I wanted to do bioinformatics, and I thought I had some of the biology and I had some of the computer programming, so I thought I could just put this together. And in 2000, 2001, bioinformatics graduate programs were just getting started in a few places. Just maybe like 2 or 3 universities offered PhDs in bioinformatics. So I interviewed at all of them. Ended up at the University of Michigan. I went there in 2001 and spent 5 years on my PhD, and ended up working on, I guess, cancer genomics, or specifically cancer transcriptomics. And I'll get into the differences of what that means in a minute. So I did my PhD in cancer biology, but basically using analysis tools to analyze molecular biology data. And then after I finished my PhD, I was fortunate to get a position as professor at Baylor College of Medicine in Houston, Texas, and that's where I currently work. I've been here for about 15 years now, and it's gone pretty well. So that's just a little brief history about me, and then I can kind of go into, and at the end, if you have questions about maybe about me or about my work and so on, we'll have time at the end to discuss those. But I'm going to go into a little bit of background in terms of molecular biology. I understand maybe not everyone on this call would have a background in molecular biology. If you get a chance to study molecular biology, I would encourage you to do so. It's a very fascinating field. There's much that we've learned about how our cells have been put together. And I'll just give some, I guess, very basic details here, hopefully enough that we can all be on the same page. But we have in molecular biology what's been called as the central dogma. And what that means is that, we have, it says DNA goes to RNA, goes to protein. And so DNA, that's like the blueprint of the cell. That's all the instructions for the cell to function, and it's unique from one individual to another. It's what makes you unique from another person, because everyone's DNA sequence is different. Everyone has a different blueprint, and so that's the DNA portion. But for the DNA for each gene, the DNA has maybe 20,000 genes. For those genes to carry out their function, they need to be transcribed, and so they need to be made into protein, and the protein is what actually carries out the function of the gene. But between protein and DNA, there's something called RNA, which is basically a copy made of the DNA, and then that gets put through some ribosomes, and the ribosomes translate that into protein. And it turns out that the amount of RNA made for each gene is going to vary, and that's what makes it interesting. It's a very important control system. Sometimes for some genes you need a lot of RNA, for some genes you just need a little bit, and so it's very important, like how much RNA you make, because the RNA then gets made into proteins. There's a lot of RNA that often gets translated into a lot of protein. Sometimes you need a lot of protein in a cell, sometimes you don't. We'll find out in cancer, everything is messed up from what has originally been designed. But that's an important thing, is to know, well, how much RNA is each gene making in the cell. And it's come about in the present day that each of these domains really represents its own field. And so DNA, the study of DNA, the global study of DNA, is known as genomics. So studying the genome, maybe how the genome is put together, how it's organized, and so on. That's genomics. And to study RNA, like how RNA programs are turned on or turned off, how RNAs are regulated. That's another field, and that's known as transcriptomics. And then the protein aspect of it is also very rich, because proteins can play multiple functions. Sometimes they can be activated or inactivated. And so we, to try to study this globally, like how the proteins in the cell are organized, that's known as proteomics. And each one kind of requires its own technology to be able to generate enough data to be able to look at things meaningfully. And we'll start off in this talk focusing on transcriptomics, but then we'll bring in like the genomics and the proteomics as we move on, as things get more complicated. But the interesting thing is, around the year 2000, when I was in graduate school, there was an explosion in the domain of transcriptomics in terms of the amount of data that could be generated, and so a lot of important studies came out of the study of the cancer transcriptome. But so I mentioned bioinformatics, and so what is bioinformatics? It's really the study of using computer science and statistics to study large amounts of molecular biology data. So now we're at a point where maybe 30 years ago, most scientific studies would just focus on one gene and study that one gene and try to understand what that one gene does. But now we have the ability to measure data on all the genes or the entire genome simultaneously, take a snapshot, a photograph of the cell and what it's doing at a particular state. So that's a huge amount of data. So how do we sift through that? How do we gain insights from that, make sense of it? Well, that's known as bioinformatics. And it's really an intersection of three domains. We have biology, molecular biology, which kind of gives us the motivation, helps us understand the system we're studying, understand what questions to ask. We have computer science. You have to deal with large amounts of data. How do you manage that or sift through that data? What tools you have at your disposal to work with those data, and statistics to be able to understand what's random from what's really non-random and what's interesting to focus on. So the statistics and the computer science are the tools we use, and then our understanding of the molecular biology gives us the motivation for what questions to ask. But I think I'm sort of you can say I'm a jack of all trades and master of none. So I know a little bit about each area. Maybe I don't go as deep as, say, somebody who's solely a molecular biologist, or someone who's, primarily just a computer scientist that would know their subject matter, know their domain a lot deeper than I do. I know enough to be able to kind of put the three together. And, I think there are different types of people working in bioinformatics, and each of these people have maybe different areas of strength. So bioinformatics, you can divide the work done in bioinformatics along the lines of algorithms and software tools and analysis. The algorithms are trying to come up with sort of protocols to work through a particular type of data and to systematically work through the data. And sometimes that takes a lot of thought to kind of understand, like, well, what's the best way really to approach the data. So that's the area of algorithms. And then there's a need for software tools. A lot of people are just great software developers, and maybe the ideas have been worked out, but they have to be implemented, and implemented in a way that other people who maybe are not computer science experts can be able to utilize them. So there's a lot of researchers working on software tools. And then there's people like me that really focus on the analysis. And I think a lot of the work that I'll show you is sort of analysis work. That's where we are actually just applying what's already been developed in terms of algorithms and software tools, but we apply it to specific questions and specific systems to try to answer a question. And so, some people can be strong in multiple areas, but maybe, so you could draw strengths from each area, but I think there's definitely a need for the analysis side or the applied bioinformatics. And that's where I work in, although you need all three. So where to begin? So I mentioned that this initial sequencing and analysis of the human genome, this is a paper that came out in Nature in the year 2001. Maybe the year 2000 it was announced and made the news. And so this was important because we had, instead of just having like some pieces of the genome being known about and cataloged, now we had the entire genome systematically sequenced from start to end. I mean, there were a few gaps in the year 2001, but they were mostly filled in, a few years later. But it was enough that we had a catalog of all the genes, and knowing all the genes, and we could it opened the door for other technology platforms to systematically measure the genes and measure not just one gene at a time, but since we had all the genes and the technology was able to grow to a point where we could measure everything simultaneously, and that means lots of data and the need to make sense and work through the data. And so now we're getting into the area of cancer omics. I showed you a slide with like 3 different omics, the genomics, the transcriptomics, and the proteomics. So ohm is like, it means kind of like we're kind of trying to look at everything. But there's different levels within the cell, right? So the DNA is a different level from the protein, is a different level from the RNA. And so, we think of cancer omics, we're understanding that there's large portions of the genome that are being deregulated, and we want to study these large portions of the genome and understand how they may be deregulated in the context of cancer. And currently, technologies will allow for the simultaneous measurements of all genes in a single experiment at various levels of molecular complexity. So I've added some more omics here from the previous slide. There's the DNA sequence will be the genomics. DNA copy. Well, normally you have 2 copies of every gene in your cell, one from your mom, one from your dad. But in cancer, some genes have no copies, some genes have like 40 copies. So that's another area of misregulation or of aberrancy. Epigenomics, that's the DNA methylation is another layer that helps regulate genes in the genome. We talk about RNA and protein, and metabolomics is another omics. Probably won't get into on this talk, but the metabolites that help your cells metabolize energy. So that's another area that's important in cancer. how does cancer arise? I mean, cancer is initiated by DNA damage. You know, pretty much every person that has cancer, there's something off with their genome. Their genome has been broken in some way. And so, and we know things that can cause cancer. Old age is putting a person at high risk for cancer, because as you get older, your genes, your cells start to misfunction and make mistakes in the DNA. So being older puts more one at risk for getting cancer. But obviously smoking, or if you're fair skinned like me and out in the sun too much, that can put somebody at risk, because the ultraviolet light or the carcinogens in cigarette smoke are going to have potential to damage DNA. And when you do that, genes that normally maintain genome integrity may become disrupted by mutation. What often happens is, you have a few key genes in the genome that they're known as the guardians of the genome. It's their job to make sure that no mistakes get into the DNA. That if there's mistakes starting to creep into the DNA, they either got to fix a mistake or they got to shut down the cell to stop it from becoming a cancer cell. But if you target one of those genes, if by if by chance that one of these important genome integrity genes becomes damaged, well then there's nothing really to stop the cancer genome from accumulating mistakes, which leads to other genes being broken and misregulated. So this is I don't know if everyone understands the idea of the cell. A cell is kind of a basic unit of life. Our bodies have trillions of cells, and each cell can maybe be a different type. Maybe it has a different form and function. Your skin cells will be different from your bone cells and your blood cells and so on. But every cell has a nucleus, has an outer membrane, has a something in between, a cytosol, and organelles, like little sort of like organs that have specific functions. And there's also an idea of pathways. There are all these proteins that sort of work together, and they kind of work in a specific sequence of events. So one protein may then interact with another protein, which then leads to another protein being turned on or turned off, and so on. So it's like a signal that's being transmitted from one part of the cell to the other. And a lot of the interaction of the cell with the outside, the cell is taking in signals from outside, and those signals will maybe bind to a receptor, send a signal to some other proteins, which then may make its way into the nucleus. And the nucleus is where the DNA is, and that's where genes get transcribed, made into RNA, which then gets translated into protein. And the thing is, in cancer, we have what are called oncogenic signaling pathways. These are signaling pathways that have a normal role in a healthy cell, in a healthy individual. But then in cancer, like some of those genes in that pathway become broken. And when they're broken, they often signal too much, or they don't signal at all when they should be signaling. And so basically in cancer, there's many genes that can become, aberrant or deregulated. And there's not just one pathway to like a cell becoming a cancer cell. Like all the pathways drawn on this slide would have the potential of maybe making a cell from a normal cell into a cancer cell if they're misregulated, or if they're become deregulated or broken through DNA damage. So even mutation or deregulation of a handful of genes can make a normal cell into a cancer cell. So it's important in molecular biology to understand these molecular pathways and how they're becoming targeted in cancer. Maybe that may lead to ideas for therapies to target those genes. In this figure is from a paper that came out in the year 2000, and I think at that point they understood a lot of the cell signaling pathways, but then these pathways, they know that they kind of send a signal to the nucleus, and then they initiate changes in gene expression. So gene expression is the transcriptomics. It's the DNA being made into RNA. and the RNA can vary from gene to gene. So these signaling pathways can cause changes in gene expression. But what are those changes? Well, there wasn't a lot of data back then, but about this time, we had the advent of DNA microarrays, which allowed us to explore changes in gene expression that happen during cancer. So that's going to get us into, still following the history of the molecular biology of cancer, moving on from the human genome being sequenced, now we're going to focus on a number of transcriptomic studies, or studies of the RNA in the cancer cell. So in the late 90s, there was a lab at Stanford that developed these, were called DNA microarrays. And what these enabled labs to do was to measure RNAs from the cell, but not just RNA from one gene. They can design these to measure all of the genes at the same time. And here's, I guess a diagram of one type of DNA microarray technology. And what you have is, you have a chip or a glass slide, and that chip would have maybe hundreds of thousands of DNA probes. And each probe would be tailored to a specific RNA species from a specific gene. And you want to know, I guess, from your sample, you want to know the level of RNAs of all the genes in that sample. So with these DNA microarrays, you could take your cell sample and take the RNA from that cell sample and convert it to cDNA and then put it on this slide. And what happens is that the probes, when the probes hits the hits a transcript for a particular gene, it'll fluoresce, it'll give a signal. And the more of that gene that's in there, the more fluorescence there is, the more light that's given off. And so, so you end up with a image like the following, where all these dots are like referring to a specific gene. And I guess the brightness of that dot, if it's very bright, it will tell you that there's a lot of that gene in that sample. And if it's dark, then there's not very much of that gene in that sample. So you could take that image and then you run it through a computer, and then it gives you a table of values for all the genes in that sample, and gives you a bunch of numbers for each gene, like how high or how much of that gene expressed. If it's expressed a lot, it'll give you a big number. Expressed a little bit, it'll be like close to zero. So, so with this technology, of course, people wanted to study cancer with it. You could study any type of cell using this technology, but of course we want to look at cancer as well, because we know that cancer is turning on a lot of genes that it shouldn't be, and other genes are off when they should be on. And so this was a study looking at prostate cancer. And what it found was this widespread deregulation of gene expression in cancer. And I guess the significance of the study, it was published in Nature in 2001. This was my advisor's lab. I was not on the paper. I joined the lab in 2001. But this is a way to kind of look at all this data together, because I think the visualization is very important. And one of the tools we use to, not just to analyze the data, but also to kind of see the patterns and show the patterns of other people, because it's important to communicate what's in the data, not just tell people what's in the data, but to show it. We have these what are called heat maps. And these heat maps, so you have a color, you have red for high, green for low. And here in the heat map, the genes are in the rows and the samples are in the columns. And the relative expression of each gene, we're looking at like, hundreds of genes, in a single heat map. So the relative expression across all these samples is shown in terms of a colorgram. So red means it's relatively high expression relative to the other samples, and green means low expression. So I guess for some of these, and then these samples are roughly ordered by normal on the left and then cancer on the right. And So we can kind of take in sections of this expression heat map and blow them up and kind of, well, maybe I want to know what genes are in here. So you can maybe zoom in on these here and maybe find some genes of interest off to the right, genes that maybe fit a particular pattern. Maybe a lot of these genes are low in the cancer, and genes here would be higher in the cancer groups. So, so there's a number of things we can take from this. We can see that gene expression profiling by itself would clearly distinguish prostate cancer from normal prostate and from this benign condition called BPH. So cancer is highly altered in terms of its transcriptomic landscape. There's not just a few genes that are turned on or off apparently, but there's like hundreds of genes or more. And so that's sort of a challenge because many of these genes may not be essential, or you can't just study 100 genes at the same time you can't design 100 different drugs, for example, to target 100 different genes. You have to know what's the critical gene here that's being, out of all these genes. Maybe some of them are secondary effects, some of them might be more primary effects. So, the problem, I guess the hope is with these technologies, you'd find some gene that's dysregulated, and then that might be a good target to design a drug for. That's worked in a couple scenarios, but it's always hard to know what's the gene to study. Now that was comparing like cancer versus normal, but you can also maybe look within a given cancer type, like say breast cancer. And anyone who studies breast cancer understands that there's more than one type of breast cancer. It's not just like everyone who presents with breast cancer, the clinician wants to know what type of breast cancer that they have, because the type that they have is going to determine what therapy they get. And this was an important study. I think this kind of validated an earlier study around the year 2000, where by gene expression profiling identifies different subtypes of breast cancer. And so, on this top here, as with the previous slide, they have what's called a clustering tree. And that's another visual tool to show these data. And what you do in a clustering tree is you let the data, so you've got like for each sample, you've got hundreds of data values for like hundreds of genes. You can let the data kind of decide how to segregate itself. So you have like all these samples, maybe you have like 80 samples here along the x-axis. And for the clustering analysis, the samples that are more like each other are going to kind of form a clique or a group. And the samples that are more different are going to be like in a different group. And what you find when you do this on the basis of the gene expression data is you get these 5 major groups of breast cancer. It's not just one big thing of breast cancer. It's like there's 5 distinct types of breast cancer. You've got what's called the luminal A, which maybe is the one that has the best prognosis. You have a luminal B, that's maybe a more aggressive kind of breast cancer. You've got an ERB2 positive subtype, which is sensitive to anti-HER2 therapy, whereby the luminal subtypes would be hormone therapy sensitive. The basal subtype, which is maybe more responsive to chemotherapy, and this normal breast-like, which is, again, a less aggressive subtype. Again, we can kind of look at the expression patterns that distinguish among these subtypes. They have kind of picked out some patterns of interest, some genes of interest, and they are preferentially high in the ERB2 subtype. One of these would be the ERB2 oncogene. And there's a therapy that targets that oncogene receptor. It blocks the oncogene receptor. Normally the ERB2 just in cancer, in some of these cancers, it just fires off and just sends signals to the cell to keep growing. Or we want to stop doing that. And so you can block it with a drug, and it'll stop doing that, and then the cells stop growing. But that only works if the cancer is expressing that ERB2 receptor. And this is getting into the era of this idea of what we now call personalized medicine, where maybe in the past a lot of the cancer patients would just get the same treatment. It didn't matter necessarily what the molecular profile of your cancer was. I think up until recently, most doctors and physicians weren't looking at that, or that wasn't really taken into consideration. But now it's understood that, well, maybe we can take a molecular snapshot of the cancer, and that would help us to understand maybe what's the best therapy for that patient, and based on their cancer genome or their cancer transcriptome. We also know that gene expression data can be used as a predictor of survival. So you can actually take a patient's tumor. So what normally happens is a patient has cancer, presents with cancer. The doctor says they have cancer. In many cases, the first thing you want to do is to operate on the patient and to take out that tumor, take out that cancer. And then you kind of hope it doesn't come back. You can give them some therapy after the surgery with the idea that that would maybe kill the remaining cancer cells. But unfortunately, in many cases, no matter what we do after the surgery and the therapy, the cancer still comes back. And when it comes back more aggressive and eventually spreads throughout the body. And so that it doesn't happen to every, so some patients will survive, have a good prognosis over the years after their first diagnosis, and other patients have a poor survival. And it turns out, when the patient first presents the cancer actually has, you can predict based on the cancer molecular data whether the patient would have a good outcome or a bad outcome, just a priori before you even follow the patient over the years. You can make a reasonable prediction. You can't make a 100% prediction, but you can make a reasonable prediction based on the gene expression data. And that's what this study shows. And we have here another heat map. Here we got the samples on the rows this time, like 300 samples. Then we got about 70 genes. And some of these samples are roughly ordered by those that had a good prognosis signature based on the molecular data on the top, and those that had a poor prognosis signature shown on the bottom. And some of these genes are, they're high with the poor, with the good prognosis, or low with the good prognosis, or inversely high with the poor prognosis. So that's that pattern, up versus down, that you can use to make a prediction and kind of assign a score to a patient, whether I think that they will do well or not do well based on what their cancer looks like now. And you can look at it in terms of a what's called a Kaplan-Meier plot, where those that have the predicted good signature, when they followed them over the years, they had a higher probability of remaining metastasis free. So you want to be you want the line to be as close to 1 as it can be, because that means that they'd have very little chance of developing metastasis or dying later on. Whereas those are in the poor prognosis signature, about half of those after 10 years had a relapse, or the cancer came back. And again, it's something you can predict years before the event. So now they have a diagnostic tool, I think, that this set of genes has gone into, where it's possible that patients with breast cancer can take this test, and the test can make a prediction. It's not always clear what to do with that information, but I guess it's more information, something that the patient might want to know. Now, here's another idea of looking at oncogenic. Again, we talked about oncogenic pathways, how these pathways can be misregulated or deregulated in cancer. And we know we have a set of oncogenes that we know about, and we can study these using gene expression profiling. And this is a study that defined what they call oncogenic pathway signatures in human cancers, and being able to use this as a guide to targeted therapies. This is again the personalized medicine approach, where they took 5 different oncogenes along the top. So they had MYC, and they had RAS, and they had E2F3, SRC, beta-catenin. These are all different oncogenes that presumably work through different pathways. And what they did is they took they took cells in the lab, and they could turn up the expression of that particular oncogene. And then when they turned it up, and they took a expression profile, they took a snapshot of what genes are high or low in that sample when you turn on that oncogene, compared to a control. Well, maybe there's for MYC, there's a lot of genes that go up and a lot of genes that go down. And so that they call that an oncogenic signature. So that's maybe, so you you do this to MYC across these different samples, that's a kind of a consistent pattern where MYC will consistently turn on or turn off these particular genes. And presumably that has something to do with what MYC does to make a cell, a cancer cell. And then they took it a step further. They could score a bunch of cancers. These are like cancer cell lines, cancers that you can grow in a lab. And we can take each of these oncogenic signatures and score these cancer cell lines based on the given oncogenic signature. And in the scheme, like red would be like a high level of oncogenic activity, and blue would be a low level of oncogenic activity. So you could predict based on the signature whether that pathway was active in that cancer cell line. And it's like the ERB2 example I talked about earlier. If you know that a cancer cell is using a particular pathway, then you maybe try to block that pathway, and then the cancer cell won't like that, and because maybe it's depending on that pathway for growth. And so they basically took the signature score, and then they also treated these cell lines with drugs that would inhibit a particular oncogene. So here in this, like in these plots here, like this top plot, they have predicted RAS pathway activity along the x-axis, and along the y-axis is response to an inhibitor that blocks the RAS pathway. And each dot is a different cancer cell line. So maybe some cancer cell lines are low activity, and so we might predict they wouldn't respond very well. And those that have high activity, maybe they would respond. If they're using the pathway, they probably won't like it if you block it. And it's a fairly, non-random linear correlation between the two. And that's what you'd like to be able to do. And you can do that based on the transcription data. I think they have another RAS inhibitor in this middle plot, and this also looks really good, a nice straight line. Maybe not every sample fits within that, but you're looking for a trend. And then you've got the SRC pathway, and there's another inhibitor that targets the SRC pathway. So the idea is maybe you could, patient is in the clinic, they're found to have cancer, take the tumor, profile the tumor for gene expression, and maybe also for the DNA. And then based on that, maybe make a prediction like what genes are screwed up in that cancer, and that may give us a clue like what therapy would best treat that patient. So that molecular information, physicians could use that to optimize the treatment for each patient. I think in breast cancer, historically, it's had the best success in breast cancer because breast cancer, you've got the ERB2 gene and you've got the ER receptor. And so if they express ER, you block that or inhibit the estrogen pathway. If they got the ERB2 gene, block that with a different drug. And we'd like to do that for other cancers. And I think a lot of institutions now are running what they call personalized medicine programs, where they have other inhibitors for other genes, but you have to know if that gene is actually a variant in that patient sample to have a reasonable guess of whether that therapy would work in that patient. So that kind of takes us through some of the early history of cancer, of these preliminary gene expression profiling studies of cancer. So this is just looking at RNA, and it's pretty rich. There's a lot to digest here. We find with these studies that hundreds of genes are deregulated in cancer, in terms of the levels that they should be at. They're either very high or very low at what the optimal levels should be. Different subtypes of cancer can be defined by gene expression profiling. Gene expression signatures may predict cancer patient survival. And gene expression signatures of oncogenic signaling pathways can be defined using experimental models, such as you can use cell lines. A lot of other researchers use mice and so on. And so, again, it's a research tool, but hopefully something that can have some clinical application as well. So where are we now? I think, in the last 20 years, we've accumulated all these data on cancers as well as other cellular systems. And what happens is a lot of this data kind of gets out into the public domain. They publish a study and they make their data available to everybody, which is great. And this publicly available gene expression profile of data represents a rich resource. So when publishing studies using gene expression profiling data, authors are encouraged to make the data available to everyone. It's just a sort of a peer pressure thing, but also journals are they kind of make that as their a lot of their requirements for publishing, and I'm fully in support of that. You don't want to just, you can't just, and then subsequent studies can reanalyze the data with different questions in mind from what the original authors had. So there's no way to kind of comprehensively look at all the genes in a single study. So maybe the authors will pick out maybe one or two genes or findings of what they could see, but it often happens that somebody else could take the same data and ask a different question and also find a very interesting result that's relevant for another question. And there are public databases that make these data available to the research community. This GEO database makes thousands of expression profile data sets publicly available. Many top journals require microarray and expression profiling studies to make the data public on GEO. And I think the European have their version of this, and it's all free and available. I guess we're going to get a little more complicated because now we're going to move from transcriptomics to like looking at other omics. And this is talking about integration, you know, different types of data, let's say protein versus RNA or DNA versus mRNA. You know, they're kind of different. You can kind of put them together in interesting ways. That's called integration. Integration of diverse molecular data types. We'll explore that a little bit here. And, you know, there's many things, and we think about integrating gene expression data with other types of data. There's many things that influence gene expression. So we know a gene is deregulated. Well, how is it deregulated? Well, there's a number of things that could be going on with that gene. Many things influence gene expression, including transcription factors, DNA copy alterations, promoter methylation, and microRNAs. I don't have time to kind of explain all of these, but just there's a lot of different things that can act on the gene. Samples can be profiled at multiple levels of molecular complexity. So that means that I can take all of these different snapshots of all of these different data types on the same cell. And I'll talk a little bit about this big project called TCGA as being one notable example. And really the interesting challenge here is to put these various types of data together in meaningful ways. And that's a lot of what I spend my time doing currently. And I think it's kept it interesting for me. So, you know, we think about, again, looking beyond the transcriptome, we think about looking at the genome. You know, I talked about DNA copy alterations, and this is something that's very much at work within cancer cells, in that their DNA copies, normally you'd have 2 copies per gene. Well, if it's an oncogene, maybe a cancer cell wants like 10 copies of that oncogene, because if you have more copies, then you've got like that much more expression of that gene. And this is, you know, this can be looked at in cancer, say, for example, using these frequency plots, where, you know, it's kind of like you're looking here across the different chromosomes. You've got your 22 chromosomes and then your 6 chromosomes. And the frequency, the percentage, over here is going to be like gain. So more than normal. So 0 would be like, I guess this is the number of patients that have a gain and a loss. So anything north of 0 would be a gain in however many percentage of patients. Anything below 0 would be like a copy loss. So the cancer wants to get rid of a gene. And what it likes to keep, a lot of oncogenes, and it likes to lose tumor suppressor genes. So the oncogenes are like the accelerator in a car, where in cancer, like somebody just has their foot on that accelerator and they won't take it off, like the car's barely down. The tumor suppressors are like the brakes. Normally the tumor suppressors just put a stop to things. They get out of control. Well, in the cancer cell, there's no brakes. They've taken out the tumor suppressor genes. Normally those are the genes that would kind of check the genome and say, something's not right with the genome. We need to shut it down. Well, the cancer is happy to get rid of that tumor suppressor gene, because then there's nothing to tell it to stop. So, and then the frequency is like, well, if it's like really frequency lost or gain in patients, that might be a clue. Like, there's probably a gene in there that's maybe important to driving a particular disease, that maybe have an important role in the cancer. You know, if there's maybe it's something that's like kind of near 0, maybe the cancer doesn't care about it. It might be a random thing, but if there's a peak, then maybe that's something like the cancer's really, I guess it's like natural selection. You know, there's lots of genome, the genome's being altered in many different ways, but the ones that kind of are preserved or continue to grow and multiply are those that have a real growth advantage. And those are the ones that are going to have like maybe a high gain for an oncogene, and maybe a loss of a tumor suppressor gene. So you kind of look for the peaks, where things are frequently lost or gained. And this is a nice study that can kind of look at both DNA copy alteration and mRNA expression. So this is this is 2 heat maps here, and this is looking at the chromosome 17 region. I talked about the ERB2 gene. This is looking at a bunch of breast cancers. I guess the cancers are going to be on the rows here. So there's maybe about something like 80 cancers in this study, I think. And on the top is like a DNA panel. So it's not expression in this top panel, it's DNA copy alteration. And red means copy gain and green means loss. mRNA, that's expression. So red means overexpressed and green means underexpressed. But if you line up like the expression with the copy alteration and look at it in terms of the genomic position, then a very interesting finding comes out where you've got a gain in DNA in this region, in this particular region within chromosome 17, and the genes in that region are also overexpressed. And one of the genes that lives there is the ERB2 gene. And there's probably a couple other genes in there as well that also have a role in the ERB2 pathway. So when you see that, then that maybe has a clue that this is kind of a convergence of these 2 types of data, maybe pinpointing a smaller set of critical genes in that cancer. And not all patients have that amplification. So you see which ones do not have the amplifications and which ones do. And the ones that do will respond to the anti-ERB2 therapy. DNA promoter methylation is another way that cancer gene expression can be changed. DNA methylation is it's kind of putting a chemical next to a gene. It's got the methylation chemical, put a methyl group in the promoter of a gene, and that sends a signal to not express the gene. So that basically switches that gene off, as long as that methylation group is there. The methylation is dynamic. So when you hear about cancer epigenetics, DNA methylation is one part of that, where it's a changeable thing. It's not your DNA is hardwired. You'll have the same DNA the rest of your life, but the methylation state from each cell that can change. And in cancer, it's changed in a not good way, because it's also aberrant, where you have genes that are silent that should be on, and genes that are on that should be silent by the DNA methylation. So the DNA methylation is another thing that's not working as it should. This is kind of sort of like looking at copy versus expression. So, this is looking at all these genes and kind of the influence of copy on the expression of that gene. And if it's like a high number, that means there's a positive correlation between the two. And so genes that are most influenced by copy alteration include ERB2, or tumor suppressor P10, which is something cancers like to lose. But the methylation is kind of the opposite correlation. So if you have high methylation in your gene, well, high methylation is silencing. High methylation silences the gene. So high methylation, gene's not expressed. Take away the methylation, gene goes on. So that's kind of an inverse correlation. And maybe not as many genes affected, but a few genes, maybe important genes can be changed by this. I guess I'm going to get into a specific example of integrative multi-omic analysis, and I'm going to maybe focus on kidney cancer. And this is where we get into this big science project called the Cancer Genome Atlas, which is this a large-scale collaborative effort that was supported by National Cancer Institute and NHGRI to systematically characterize genomic changes that occur in over 10,000 human cancers. So I think the data sets we had up until the Cancer Genome Atlas, which started maybe 10 years ago, I think were, you had a lot of interesting data sets, but this was like the mother of all cancer genomic data sets. And it's still being heavily used today because it was a big, so it's what you call big science, right? A lot of resources, a lot of effort put into this one project. Kind of have like a big comprehensive data set for everyone to be able to go to. Kind of a big vision type project. And I think in many ways it was quite successful. But now we have all this molecular profiling data on 32 different cancer types in TCGA, and not just gene expression, but also the genomics and some protein and epigenetics as well, all in the same samples. And there are a lot of high-profile papers that were published on these data. I mentioned there were 32 different types of cancer, so you don't have just breast cancer or just prostate cancer. We've got all of the major types of adult cancers represented in this project. And for each cancer type, basically TCGA would get a bunch of scientists together, and those scientists would go over the data. I was one of those scientists, and we would make some findings from the data, and we publish a paper on it, on that cancer type. And I think I think they published probably 20, close to maybe 30 marker papers, what they call marker papers, which is like papers focused on a particular cancer type. And I think of the papers shown here, I think I contributed directly to 4 of them. So I was able to make a lot of contributions to a lot of these high-profile papers, even though I was just one team member. This is what's known as team science. And nice thing with all the different cancer types being represented here is that we could do these what's called pan-cancer studies across disease types. So this is like, here they have like 12 different cancer types, and then like 5 different molecular platforms. They've got copy number alteration, DNA methylation, microRNAs, which is another type of RNA, mRNA or messenger RNA, and then protein or RPPA, which is green. So each platform has its own color, and what they do is they kind of take the information from the different platforms, and then they put it together, and they have each platform have a say in what the subtypes of cancer should be. And they put all these different cancer types together, these 12 different cancer types, and they just kind of let them segregate on the basis of the molecular data. And they mostly segregate on the basis of the tissue of origin, which wasn't surprising, but I think there are also some interesting variations on that. A couple of surprises came out of this study. But the whole point of this slide is to kind of introduce the notion of pan-cancer studies, where, before we just kind of did studies on one cancer type at a time, and then you have people that really know their type of cancer. You have people that study breast cancer all their life, and they don't really look at any kind of other types of cancer in their research. But now we're at a point where we've got data on all these different cancers, so much data, there's nothing to stop us from just putting it together and looking at everything and getting a snapshot of not just one type of cancer, but all the types of cancer together. See what are commonalities across different cancer types. What are some maybe what makes one cancer distinct from another type of cancer. So the point here is not to get people lost, but the data are just getting bigger and bigger, and the questions are getting kind of more and more comprehensive. Last thing I'll talk about, I guess I'll talk specifically about some of my work from several years ago, and this was looking at this TCGA-led integrated analysis of renal cell carcinoma. And so this is where we're going from just RNA data to kind of looking at all the types of data together. And so cancer genomics is, again, this team science endeavor. So here's the author list from this TCGA paper on clear cell renal cell carcinoma. And I guess I'm one of them. But there's but the author is like, just this one consortium author. But then there's these individual authors within that. I'm one of them, along with 345 other people. And some of those people were, say, physicians or clinicians that collected the tissue samples. They had, they're just automatically on the paper for helping collect the tissues. Other people just generate the data, and that's all they were involved with, was data generation. And then there were people like me who did the analysis, took the data and worked with others and try to kind of find something interesting or new in the data. And so this was one where I actually helped to lead the analysis with someone else. And so I had a pretty heavy role in this paper. And I'll go over some highlights and try to understand some of the things that we could learn from these data. So I had a role in kind of helping coordinate the analysis and helping to put the manuscript together and to help write it with others. So we can look at carry out gene sequencing at the DNA level and find what are the significantly mutated genes. So the DNA, you can have a base that's changed, or maybe multiple bases that are changed in that gene. And so, and some genes are frequently mutated, which kind of indicates that they would have a role in that disease. And so you kind of get a short list of significantly mutated genes involving this cancer type. Here, we're looking at like 385 kidney cancer samples, or this is known as clear cell renal cell carcinoma, or CCRCC. So I'm looking at like 385 tumor samples along the top here. We've got like VHL, one of the top mutation hits, very important gene involved with metabolism and PI3 kinase pathway and so on, and other like chromatin inhibitors and so on, and couple key gain or loss events. Also methylation. So the VHL sometimes it's not mutated, but it could be methylated, where basically that's like you're turning it off. You can maybe turn it off by having it mutated or broken. That's one way. Another way is to have the methylation increase to where it's not expressed. Either way, you're taking out the VHL gene, which is an important tumor suppressor gene in this disease. It's pretty high. It's lost. Like maybe one copy is mutated and the other copy is lost. So blue is lost here in the 3p21.1. That's where the VHL gene lives. So that VHL that's something the cancer wants to take out. This type of cancer. Other cancers take out something else, but the kidney cancer samples wants to take out VHL. And then we have these other genes that are like these regulators. They have a specific role as chromatin regulators, and the chromatin kind of helps organize the DNA, maybe helps it to fold properly and so on, so that's also deregulated, in this type of cancer. There's an important oncogenic pathway called PI3 kinase pathway, and that's also being taken out in some of the samples with taking out a couple of the breaks of the PI3 kinase pathway, as well as gaining a couple of oncogenes in the PI3 kinase pathway. And this is looking at DNA methylation. So this is another color scheme. It's another heat map, but they found out, one of these chromatin, one of these genes involved with the chromatin is the SETD2, and if you mutate that or take that gene out, there's lots of changes in DNA methylation, and a lot of genes that are like under methylated or over methylated, as compared to like the other tumor samples that would have like a normal SETD2 gene. So you can see changes, like you change one gene at the DNA level, then it has downstream effects, and it can affect globally the methylation and likely other things as well. So that's something you can see if you have these different types of data. You can put together the DNA mutation data and the DNA methylation data together, kind of see that's how a change in one gene is going to affect a change that we see in another data type. So that you can start to maybe line up the two patterns and kind of see like a cause and effect, or correspondence at least between them. And then this was analysis that I was involved with in defining molecular signatures and pathways of aggressive CCRCC. And this is similar to that breast cancer study I showed you, where you could define a set of genes that were associated with a better or worse outcome in patients, and make a prediction in another set of tumors. And that's what we did here. But here we're not, that breast cancer study just looked at mRNA, but we have these other data types. So we can look at microRNA, we can look at protein, we can look at DNA methylation, and all these levels of data have some information about what distinguishes the more aggressive cancer from a less aggressive cancer. And that's something we'd like to know, because maybe there's something that's driving the more aggressive cancers that we want to maybe stop, because we don't necessarily understand fully why some patients have a good outcome and why some patients don't. It's not always clear. So we can find some molecular drivers of that aggressiveness that may give some ideas for pathways to target. And this is where we had the individual genes, and then we wanted to kind of put it together, get a big picture of it. It helps to understand molecular biology and understand what's been studied about these pathways over the past 30, 40 years. And so I worked with a couple of very good molecular biologists, and they helped me draw out these pathways, these metabolic pathways that have been well studied over the years. And we've got all these genes in this pathway, and some of these, we've got a color on each gene, and the color is the correlation with the patient survival or the patient outcome. Genes that are red were associated with a worse outcome. They'll be high in the patient that did badly. Genes that are blue are those that are associated with a better outcome. And so those are genes that are low in patients that did badly, or alternatively high in patients that did better. And the blue and the red, the relative changes, kind of means a lot, because some like hot regions where there's some key red genes, and some blue regions where there are some like other genes that are just low with worse prognosis. And so what this kind of shows is this what we call a glycolytic shift, or a type of Warburg effect. So like, in the 50s, I think, or a long time ago, someone named Warburg theorized that the altered metabolism can be driving a lot of cancers, and that they need like a lot of glucose, but at the same time, so they, this glucose pathway is really hot in these aggressive cancers. But then, they want to like dampen things like the Krebs cycle and the mitochondrial pathways. So they basically change the way they process energy, and so they need a lot of energy, a lot of quick energy, and so this glycolytic shift is the way they do that. They got to change their metabolism to kind of keep their aggressive pace of growth. And so this is another level of change, a level of pathway changes, where we're not just looking at individual genes now. We're kind of putting it all together into like a big picture, and using our domain knowledge, using what's been studied about these pathways to really get a global sense. And I think having the big picture view of the altered cancer cell with all these different types of data at our disposal can get us a much bigger picture than what we've been able to get before. And so that's just an example of the ways that we can put these different types of data together. It's getting more complicated. I mean, the data sets are getting bigger. There's people are figuring out different ways to kind of creatively put these types of data together to answer different questions. And so that's what kind of, for me, makes it a very exciting area to work in. And so hopefully that will give you maybe a taste of where the field is at and why I find it interesting. And I think that would probably be a good place to pause and I guess see if there are any questions, I guess about my work, or about how it is to do research, or Mindliner research, what's that like on a daily basis?
Yep, there are a bunch of questions.
Okay.
And well, thank you, Professor Chad, for giving us this like perfect overview of cancer genome analyses. And I think our participants learned a lot on how these analyses are carried out, and the main applications, because there are very vast applications of this field of research, not only for cancer treatment or cancer drug discovery or drug development, but also could be also applied for cancer prevention, if we could really like pinpoint the exact pathways at which the genes get aberrant, or where the problem arises.
Mm-hmm.
But we have a bunch of questions from Ma'am Leonora Henniston. I think she's here on Zoom, and I think she wants to ask her questions live.
Okay.
Maybe just acknowledge Ma'am Leonora.
Hi, good morning. Good morning in the Philippines.
Good morning in the Philippines. Yes, ma'am.
Good evening in the US. Yeah.
Please go ahead.
Yeah. I'm very excited to talk to the actual author, to the author of a certain paper, and I think I will learn a lot. My first question is, what software was used to show the relationship of each of those genes done by Dana Sekaran? Is it a free download?
Oh, this slide here, this pathway slide?
The first, I think the first few slides, the nasa Karen.
Oh, like the heat maps?
Yeah, the heat map. Yeah.
Huh. Well, a lot of bioinformatics people use a software called R, which is used a lot in statistics analysis. So a lot of people use R to generate heat maps. There's another software called Java TreeView, and I use that a lot. Java TreeView, and I think a lot of those early slides used an earlier version of Java TreeView. I think they just called it TreeView, and then someone made a better version and called it Java TreeView, and I use the Java TreeView.
Okay.
Yeah.
Another question. Is there a database wherein those genes that were presented by Sorley be attributed to show that those genes are in a certain metabolic pathway? You know, some metabolic pathways are very complex, and most of those genes are, we don't know which are expressed and which are poorly expressed.
Yeah. So how to get like a pathway database, you say?
Yeah. There's one?
Yeah. Well, there's a few, and I use so there's a database called the Gene Ontology, or GO. And that kind of like categorizes all the genes in terms of their functional groups. And that's very useful, you know, like all the ribosome genes, they'll categorize them as ribosomes, and the receptor tyrosine kinases, that's another group. And there's different levels you can describe each gene using a controlled vocabulary. So I use a lot of Go term analysis. I think I published some software on that in quite a while ago called SIGTerms, SIGTER. MS, but other people use a software called David, and like the name David, and other people use something called GSEA, and they're all kind of get at the same things. Some of them will have like the Go terms, which is good, and some of them use like other like pathway related gene sets. Another useful resource is the Wiki Pathways, so WIKI Pathways. Because that's got like where like I think like this kind of pathway, there's a version of that on Wiki Pathways that I refer to quite a bit. So a lot of the metabolism pathways. I think we did a study on COVID-19 virus last year, and like interferon pathways are hot in infected cells and so on. So that's just kind of a nice resource because it's like kind of drawn out, like because maybe the Go terms doesn't tell you like the sequence of events like a pathway is supposed to represent, but the Wiki Pathways is kind of drawn like what I show here on the slide.
Okay, I think this is the last question to give others a chance.
Okay.
Regarding this last slide, the survival correlates, when I observed that you analyzed for RNA and protein, and they were tagged in a specific certain pathway, and you were able to analyze it as worse outcome and better outcome. Does it mean that RNA was analyzed on one hand and protein was analyzed on the other hand, and you plug them in here in the metabolic pathway?
Yes, we analyze those independently, and as I think in TCJ there are like a set of maybe 200 proteins that are also profiled, and some of those proteins are like they're like phosphorylated forms of proteins, which you don't see at the RNA data. So the protein's different, and sometimes RNA and protein don't always correlate. You can't always take it for granted that if you have a lot of RNA that you're going to end up with a lot of protein. Generally you do, but sometimes there's a dichotomy between those. And I think now we're getting to the point where there's lots of proteomic cancer data being put into the public domain with another big science consortium called the CPTAC. And I've got a couple papers using CPTAC data in the last couple years where you maybe look at protein and RNA, and sometimes they don't always agree, or there's things that we see in the protein that we could never see in the RNA data. So that's just another layer, another level to look at. And I think with TCJ, they just had like the platforms are designed just for 200 proteins, and now you've got data sets of like thousands of proteins, close to 10,000 proteins across all these cancers now. So that's another thing to look at.
By the way, is it okay to have a copy of your paper, because some of these are usually hard to download. You don't have a subscription.
Everything's published, I've shown. I'm happy to share it. I don't know. Share the slides, you mean, or?
Yeah.
Yeah, I'm happy to do that. I guess I can talk to the moderators how to do that.
Yeah, I could probably provide Ma'am Leonora some copies of the articles. Chad, can you just go back to the slides with the publications, and I can just like screenshot.
Oh, I'm sorry. Oh, I see.
The last one.
This one here?
I think this.
Okay, yeah, that's the paper
Yes.
one over. Yes.
All right.
Okay.
And I'll just provide you, Ma'am Leonora, the copy.
I have some data in proteomics, because I have my dissertation is about proteomics, but I think there are seeing your data. I think there are a lot more how to interpret the data I have.
Okay.
That's fine. Very interesting. Thank you very much to give others a chance to ask questions.
Thanks for the question.
Thank you, Ma'am Leonora. We have another question here from June Alexis Santos. I don't know if he's around here. Maybe he could ask his question live, because he's. Well, I'm just gonna read the question. Hello, Dr. Creighton. I am a graduate school student working on viral proteomics and structural bioinformatics, and yet considering myself not yet well versed in the field. Just to have an idea of what bioinformatics can offer in the field of cancer therapeutics development, I just want to ask, what is the role of bioinformatics for cancer therapeutics and other cancer treatments, such as chimeric antigen receptors treatment?
Yeah, I think a lot of along the lines of some of the studies I showed. I think you profile like maybe hundreds of cancers now. Try to get a sense of like what's important. You want what's called the landscape, I guess, the semantic landscape, like what's changed. And and with many samples you can say like, well, statistically, like what's most frequently happening, and what are the subtypes of the cancer. So I think each GCJ paper tried to do molecular subtyping, like the breast cancer study, to try to like, okay, these are the subtypes, and then maybe you want to find like the mutations associated with each subtype. And I think with that information, then maybe you'd have, instead of like all 20,000 genes, you would have a much shorter list involving that cancer type that would be in a personalized medicine setting that you'd want to focus on for a given patient's tumor. Like you wouldn't necessarily try to test everything. You would have an idea.
So you just could narrow down the analysis.
That's right.
To make it more targeted.
Yeah. When you take it to the clinical setting where it's not research, you just want to be able to run a test and know what a few genes to look at, and so on.
Right. I'm just curious about these analysis, because you can analyze like cancer genome at different levels. Is there like a systematic way of looking at it? If you could start with just DNA at the DNA level, or RNA, or is it mostly done like simultaneously at various levels?
There's more than one way to do it, and I think a lot of people maybe will work off of previous examples. So maybe it depends on the question. I think with subtypes, the expression data, like either protein or RNA, is a good place to start to find the subtype. But some of the slides I showed, they actually put all the platforms together. All the platforms kind of had a say. It was very democratic. So each platform got an equal say, and so you could do it that way. But maybe oftentimes, like maybe you want to maybe start with the DNA data to kind of see what's the mutations, and then see if that corresponds with the change in expression. So I guess there's just not one single path to approaching it. I guess that's sort of what makes it interesting. It maybe depends on the question.
Yeah.
So I actually have a question. This is actually so, Chad. This is actually more of a mentoring type of question for our students, right? For them to
Mhm.
appreciate. So I actually like the fact that you showed this particular slide, right, where you guys are dealing with large data set, right? And in order for the development of this area to progress and to be accelerated, the sense of community is very important. Could you actually speak more about it, about like why it's very important for scientists to collaborate to actually progress like some of these
Yeah.
activities?
Well, I mean, most of my, probably like 90% or more of my work is collaboration.
Yeah.
There's a few studies I can just do that are just writing a piece of software, for example, that's just me, or a couple studies like that. But I think pretty much everything I've done, even my own papers that I've led and written myself, involve other collaborators, because so there are different types of studies. There are some of these like genomic studies, like the TCGA studies, where I don't have the capacity to generate the data. And so that was very much a big team effort. But also I work with a lot of molecular biologists who just, like I was 20 years ago, and they're working in the lab where their students are, uh, running experiments and generating data, and they need help looking at the data. So a lot of the papers I publish are just me helping the molecular biologists with one step in their project, where they need a data analyst, and I can just be that person to help them with that part of it. So I can play a role. I like both types of studies. I like to. It's fun to come up with your own idea and write your own paper. But even when I'm doing that, I'm taking data from the public domain, which a lot of people's efforts. And you do that, you try to understand like what's been done, and you're kind of putting yourself as part of a community. Even when you're writing your own paper, it's like you're kind of part of this organic network. You got to talk about like how your study fits in with all the other previous studies and how it's taking it forward. But even in a real sense, I probably collaborate more than many other scientists, and that's kind of my primary role here at the college, is to collaborate. So I haven't done as much of this big team science. I mean, that was kind of a special thing. I did that for 10 years, so that was a while. But and I'm still kind of so a lot of the teams I'm on now are maybe smaller, but there's a fair number of Zoom calls, and it's still highly collaborative, because I just run. I have what's called a dry lab. I have a computer, and I'm doing analysis, but if there's no data, there's nothing for me to do. So that's, you need wet lab biologists, people actually in the lab or collecting tissue samples, running, generating data, and that's how we work together. So definitely, in this field, you definitely want to be able to work well with others. So I guess that's a soft skill, but that's a skill we all have to have.
Yeah, and to be honest, Shad, I think the reason why I asked that is because that is a very important aspect of doing work that I believe is actually not very well practiced in the Philippines. And I think it's very important for scientists like us to also like foster that value, that for example, like for us Filipinos who are practicing science abroad, we actually get to do more because we're part of cross functional teams. We're part of an interdisciplinary team of scientists like working towards providing solutions to pressing problems. And I'm hoping that the Filipino students and teachers out there who have actually heard what Chad had shared and has seen like some of these outcomes of collaborative programs, I'm hoping that you actually see the value in collaborating. So you don't actually have to always work on your own. There really is like value in tapping into other people's expertise. So you don't have to be an expert in everything as well. You just have to be able to look around, identify people who actually are experts in certain aspects of your study, and work together. There really is like some value in there. And then another question. This is like for the high school students, Chad. So what particular STEM courses or areas should they actually specialize on, or like should they train? Should they have like some fairly strong training for them to be able to say work in cancer genome analysis? Biostatistics is actually one of them. But what other allied areas are actually involved in this field?
Yeah, it's funny. Like I said, for my bachelor's, degree in my undergraduate, I had no plans of doing bioinformatics, so I majored in physics, which I don't use now. And if I had to do over again, I'd major in statistics and computer science because I'm using that.
Yep.
And so I don't know, like I didn't prepare for it in the right way. And I guess because it wasn't there, yeah, I think, yeah, so I use a lot of computer science and statistics now, and so that's always good to learn. But they always, I guess they push computer programming a lot, and so everybody's studying it, which is good. But I think the physics was very good for me. I think because you have to think conceptually, and I go into the physics because I like math, I like calculus, and I like kind of the puzzles and problem solving. It was just a different time. Like I did, like everything was done on pencil and paper. Yep. Like 99% of my undergraduate work was just working out problems. I don't know, I debate if that was like the best way. That's just, but you do a lot of problem solving. So I guess anything you do, I still think math is really important because I don't think it's, because I think the computer science is actually pretty easy to pick up if you're,
yep.
if you're a good analytical thinker. Like I was able to pick it up, and no problem. And the biology, well, a lot of it's memorization, but I think that the math is really good. But also, I think I read a lot as a kid and well into after college. Like and so much is written communication. Like if you can't, you gotta communicate yourself in writing. And I have kids now, and I'm just encouraging them, I guess, on their free time to read as much as they want. We'll buy them all the books they want to read as long as they're reading, because I think that's STEM is not just science, and I think a big part of it, you want to be able to communicate in writing. Otherwise, who's going to hear about your ideas? So that's a big part of what I do now is a lot of writing. And that's what scientists do. Like a lot of them write a lot. So it's an important thing too. So it's all important. I think I think everything you learn, there's something to learn from it. Everything you're coming across in school, all the courses, there's always something to learn from it. So I just go into everything, I think, with enthusiasm.
All right. Yeah, thank you.
And there's really just no substitute for reading.
Yeah.
Yeah.
Exactly.
So, I hope all of our students joining here today just realize the importance of reading, because now it's just all online stuff and webinar videos.
Yeah, and I actually like the fact that you shared your story, right? That you didn't really chart your future in such a way that, okay, like 10 years from now, I'm gonna be a biostatistician. Like you started in physics, and then opportunities, one opportunity led to the other that like eventually landed you in biostatistics. But I guess I also like the fact that you said that, even though you're not practicing physics anymore, you also actually, like, it's a synthesis. It's a synthesis process, right? How you make sense of numbers, how you make sense of like the data that the software actually spitting out. So this is also like to share with our students that it's always very important to challenge yourself. And I'm sorry about that. It's a friend of mine calling. Anyway, so yeah, it's very important for you guys to keep pushing yourself. Like if you get presented with some opportunities that may be out of your comfort zone, it doesn't hurt to actually take risks sometimes. So I think that's what I actually like about Professor Chad's story, like that he shared like in the beginning of his talk.
Well, this is another thing. I don't know. In 20 years, I don't know what the next thing is going to be. Like this is what I'm doing. I mean, my kids, I don't know what they're going to face. There could be something new that comes on that's like really cool. And I guess that just like they have the analytical tools and ability to kind of learn things and to pick up on things, then I think they'll be in the best position. You can just go in a direction. You want to kind of go and just do the best of whatever you're doing. And, I think an object that's in motion can be easily steered in another direction, whereas something that's just like not moving, it's like stuck in the mud. There's a lot of inertia. Yeah. And so it's important just to have a goal, be flexible, but just move towards something. And you can be open to kind of things as you know, as new things come up or maybe the situation changes. There's lots of options.
Yep.
Yeah, and
And I
it's
guess like
important.
yeah.
Yeah, to find your niche probably, because
Yeah.
Chad kind of like figured out on the fly what he liked doing. So
Yep.
so it's really
And I
important.
have to say, students now have much better access to resources. So that's something that we encourage all you guys to do, keep exploring. There are materials out there. There are even things that you can learn without actually having to you know go to a formal school. So
Hmm. Yep. So I think we can wrap up the discussion at this point. Doc Jeffrey, do you have anything to share to us
yep,
about
just like
our?
a couple of announcements of some events. So Chad, I'm gonna take over the screen.
Sure.
Don't get surprised if you're sharing. Okay, actually stops.
Hmm.
Okay, so just a couple of things, po. So April 30, 6 days from now, I think in the Philippines, it's the 24th already. That's gonna be the deadline for submission of entries to PhilSci Hub Research University's Lions Den. So this is the Battle of Science Investigatory Project Ideas. So submit your project proposals. The instructions are actually on our Facebook page and on our official website. Okay, and we have a couple of upcoming webinars. So over the past, the past three webinars and this upcoming one are actually all focused on um,
Data
computer-based
analytics
research, essentially data analytics, computational
methods.
sciences and
Hmm.
methods. Okay, so we do have this computational chemistry and its application special topics webinars, essentially how computational chemistry is applied in research or is used in research. This is going to be delivered by a team of experts from the University of the Philippines in Diliman. So we have Joaquin Gaza, Almira Ocampo, and Professor Ricky Nelia. So this is going to be on May 8th, 2021, this year, of course, at 10 in the morning in the Philippines. So this is a live Zoom and live event. So for those of you who are interested in learning more about computational chemistry, at dun sa mga hindi po familiar pa, check this out. And then after that, on May 15th, I will be delivering PhilSci Hub 6 research training course. This focuses on science communications. I will be providing you with some guidance as to how to write a scientific paper. In that talk as well, in that webinar as well, JP Onya and Marty Mateo will be joining me. So they'll talk about how to effectively write laboratory reports and also the importance of science communication. So watch out for that May 15th. And then on June 5th, Professor Dindy Voiles will be returning. She will be introducing you to the world of alkenes. And also in this session, I will be participating by introducing all of you to the basics of organic reaction mechanisms. So Professor Chad mentioned methylation earlier, right? So that's a chemical transformations. Like we're gonna start to introduce you to all these organic transformations. So watch out for these events. You can sign up for these, by visiting our website and our official Facebook page. All right, so I guess that's all that I have for now. And we have a couple more of other webinars that are actually up and coming. We have one on sustainable chemistry or green chemistry. Is it green chemistry, JP?
Yeah.
And then we also have a seasoned professor of chemistry who will deliver a lecture third week of May. That's about inorganic
Yeah.
compounds, or that's somewhere
Yeah, we
somewhere.
still have to sort out our calendar because we
Yeah,
have a lot of activities coming
yeah,
up
we have a lot of activities extending over January
next year.
2022. So Dindy Boyles did a great job of recruiting a lot of seasoned scientists and experts, like who will share their expertise and knowledge. So yeah, that's mostly it from my end. And yeah, I mean personally, I would like to thank Chad for lending us his time. So it's already 9:50 here in Houston. So thank you so much, Chad, for being very generous with your time and expertise.
Oh,
Yeah.
glad to do it.
And I would just like to present this certificate of appreciation to Professor Chad Creighton of Baylor College Medicine, Baylor University, for delivering the webinar Cancer Genome Analysis, organized by the Filipino Science Hub and held via Zoom and YouTube Live on this 24th day of April 2021. Signed, Dr. Jeffrey Bunkin, our PhilSci Hub founder and CEO, and myself, JP Onya, Head of Research. So Chad, in behalf of the PhilSci Hub family and the PhilSci Hub community, the research community of the Philippines, we'd like to extend our deep gratitude to you for this very rare opportunity to share your expertise and really come in close contact with our scholars here in the Philippines. Well, they're in the Philippines. So thank you very much. And at this point, I could probably share the link now for the certificate.
Can we perhaps have a picture with Chad?
Sure.
Yeah, Chad, if you don't mind, is it okay if we take a picture with the entire
Sure.
participation? We actually had like a full house on Zoom, and we have about like a total of about like almost 200 participants like for this event. So you've touched a number of lives in the Philippines. They now know like cancer genome analysis and more a few more things about molecular biology. So thank you so much. Who's gonna take a picture? JP, could you take the pictures, please?
I can take a. People can probably turn on their cameras at this point. Okay. Ready? 1, 2, 3. Okay. One more. One, two, three. We have 4 panels here. So.
Yeah.
1, 2, 3. Okay.
All right.
1, 2, 3. Thank you.
Yeah, thank you so much, everyone. Chad, thank you so much. We're hoping that at one point, sometime in the future, we can invite you over again for another event like this. Thank you so much.
Yeah, it was a lot of fun.
Mm-hmm.
Yeah.
Yep.
Thanks,
So I
everyone.
posted the link for the certificate. I mean the evaluation form for the certificate issuance. So I hope everybody already got that. And for those who will be watching this recorded webinar video later, the link for the evaluation form is posted on the video description in YouTube. So you can just like scroll through that and fill up the form. Please wait for 7 days to get your certificates of participation because we're issuing in batches. So don't worry if you don't receive your certificate soon enough. You're gonna get it after 7 days. Yep. So I think on this queue, Ma'am Dindi, do you still have any other?
No, I just really want to thank Chad for talking tonight. Yeah, we are so very fortunate to have you. We've learned a lot.
Yeah.
And perhaps we can invite him to just like a 10-minute debriefing after this, Chad. We will be sending a separate Zoom session.
Oh, okay.
Yeah, if that's okay. Thank you so much.
Yeah.
All right.
All right. Thank you very much, everyone.
Bye.
May you have a very good weekend, and we hope to see you next time for our next webinar here at PhilSci Hub. Thank you very much.
Thanks, everyone. Take care.
So I'm gonna end the session now. Maraming salamat po.